Sensors, diagnostic, imaging-guided nanomedicine, targeted drug delivery. I do have the honor to chair this session. My name is Christopher Lexer from Erlangen. The first speaker today is Professor Dr. Med Dong Soo Lee from Seoul, and he's talking about intrathecal exosomes, brain imaging for CSF-lymphatic, Efflux, and Neuroimmune interface. The floor is yours.
Thank you. Good morning. Upon the title, I'm going to combine three issues here. The first one is The first one is CNS system immune system interaction or interface. The second one is extracellular vesicle or exosomes, mimetic nanovesicles we used as a plausible surrogate mediator between the CNS and outside. And using our technology of CSF drainage imaging, we just developed using copper-64 albumin and exosomes PET. So the first one is the second one, the exosomes mimetic nanovesicles in one slide. There are so many problems to solve in extracellular vesicles in physiology and diseases in vivo. So a few among many more, if I just list it, specificity for target cells and tissues on organs of those extracellular vesicles on in vivo studies. According to the Matthews excellent review a while ago in Nature Cell Biology, after reading all those specificities issues, I found that there is no information regarding the specificity of secretion or uptake. So meaning we don't know anything about that. And how the extracellular vesicles are working on adjacent tissues or adjacent cells or remote ones. If I just adopt an analogy, cytokines or hormones, it's paracrine and endocrine counterparts. And how long is it acting? And EVs are considered, have been found to be produced by every cells on Earth, from prokaryote to eukaryote, of course, the humans. So EV diversity is huge. So related with their sources, do we know about the biodistribution? No, we don't know. Beside non-existing technologies for single exosomes are a million times smaller than cell. So single cell, the proteomics and transcriptomics are on the horizon, on the market, actually everywhere now. But single exosome studies are beyond the current situation. Then the second issue, the neuroimmune interface. So everyone knows that there are barriers between CNS and outside of CNS. So here, I just pay attention to the outside of CNS immune system. Blood-brain barrier is famous one with a Claudin 5 between brain endothelial cells. So because of that tight junction, the astrocytes are forming the glia limitans using their foot process just beneath the pia. They don't have any tight junction. So meaning there are space between the endothelial basement membrane and also the glia limitans. So we call it perivascular space, and perivascular space is filled with in physiology, filled with the CSF fluid. And then there is another barrier, brain and the outside. Brain and CSF does not have any tight junction, as I just told you. PIA has just a gap junction. And there is a arachnoid barrier cell. They're having a Claudin 11 to make a tight junction here. They called it tight junction. So that kind of the tight junction was trusted by scientific community for such a long time. However, recently, a while ago in pigs, they were found that cranial arachnoid granulation-like dural gaps. Sometimes they call it fissure here. So that arachnoid barrier cell layer were suspected to not to be perfect. And in very recent paper in June in Science, the Swedish team has reported, interestingly, arachnoid barrier cell layer has a kind of a leak, the breakpoint, while just looking at the subarachnoid lymphatic-like membrane structure, very subtle structure. So that paper was paying attention to that newly found SLYM, slim. But in their paper, I found that in rodents and humans, there is another the kind of a leak of the barriers. And also, that dura is now trusted as a kind of having lymphatic structure inside, not just a kind of a thick supporting structure, but a very active site. And bone marrow, the vein, bone marrow sinusoids, sinusoids has a connection according to that paper using V-disco. And that channel between bone marrow and the dura, venous channel, is being used by lymphoid and myeloid cells. So leukocytes commute between the bone marrow and also the dura. And dura has a arachnoid barrier cell layer, which has a kind of a leak, meaning everything is connected. So that means that ovalbumin injected here to the CSF space, that ovalbumin can come out to epidural, the epidural networks, lymphatic vein networks, and which was proven by this V-disco study and also the contrast dye, MR contrast dye. So in sum, there is a transportation checkpoints between CNS and outside here. It should be tight junction. However, there is a leak. So the skull bone marrow is venous sinus is connected, and then CSF is also connected. So we suspected there might be a kind of a good method to visualize that. So keys to the success of the imaging, the distribution of intrathecally injected albumin or exosomes mimetic ENV, nanovascular vesicles here. The first one was the click reaction. At first time, I just developed the technetium HMPO to visualize to label the extracellular vesicles. That technetium HMPO is inside, just like cells. However, in this case, the membrane is being labeled using the famous click reaction, alkene azide reaction. And then the PET MRI, and we used E. coli-derived ENV and also the mesenchymal stem cell-derived ghost ENV. Ghost means does not have any inside material currently. So after looking through all those literatures for the last decade, we just adjusted the smallest volume. However, the enough volume to just visualize. Intrathecal injected one is going up to the basal cistern, and then they visualize the entire CSF space, and then whereabouts. This was the recently published one. At first, we just labeled copper-64 to the albumin and injected, interestingly, here, superficial cervical lymph node, which is also called submandibular lymph node, and here, but faintly, but definitely here, deep cervical lymph node, and interestingly, iliac lymph node and sacral lymph node. Entire CSF space is visualized until 24 hours. So what about those the systemically drained the radioactivity goes? It's according to this ITLC, we could just find that albumin is degraded until 24 hours, and then it is going to be excreted via urinary bladder. And we just simply compared aged animal and also the adult. And we could find after initial perturbation, there were much overlap, but after 24 hours, it's definite to show the age-related decline of till how many minutes?
5 minutes.
Yeah, 5 minutes. So, there is a definitive difference between, and please pay attention to this, the inter-subject variance. There is no inter-subject variance, which was the exciting and stimulating and interesting. So, initially there was a kind of this amount of one is just 1/6 of the entire volume of the CSF, and which seems to be ameliorated from the initial perturbation. The next day, the CSF spaces still have a radioactivity and releasing, and then lymph node is recovering. So that was the story. And we just adapted this method for ENV visualization. At first, E. coli derived ones here. And we could find, so actually we could differentiate the liver uptake here by just a serial imaging. There was a prominent spleen uptake and gradual the bladder release. However, liver activity stayed. So we suspect that copper cells, however, small amount is in the intestine and finally in the rectum. So that I call it the mostly copper cell here, and that kind of labeling did not need any kind of separation procedure. So initial ones were here using the another mammalian or eukaryotic mammalian or human derived ENV. It's been taken up in the liver but after IV injection. Gallbladder is shown, and many intestinal activity, and finally rectum. So I can say that the liver activity is just hepatocytes, and that's the contaminants because we did not include any separation procedure. So while having this kind of technology, we've just looked through, and in this famous paper 2 years ago, they have radiated body bone marrow or the brain bone marrow. They could find segmental match between the brain and skull bone marrow and spinal cord and vertebral bone marrow. In immune system, bone marrow is central. In the nervous system, brain and spinal cords are. So I just thought, what about the sentinel watch from the side of the lymph node to look into the central nervous system? So immune system, recently, there comes the more and more evidence that the tissue-resident entire immunocyte systems are everywhere from the birth, from the yolk. And so now intrathecal injection. I'm going to show last two images here. This is the first one, E. coli-derived ENV. You can see even the tail, coccyx, and then the injection site, superficial cervical lymph node. That's cistern brain, and then deep cervical lymph node, and here iliac, and also there, and then sacral. Interestingly, mesenchymal cell-derived exosomes did not show anything. And interestingly, these are gallbladder and here, and bladder. Superficial lymph node was a little bit shown. And unlike humans, the mouse has a nasal cavity drainage. So we suspect that. So, summing these image readouts, lymph nodes are showing the specificity by source. Liver, also specificity by source. And spleen, only for E. coli-derived ENVs. Yesterday, in another conference, another session, I found that liposomes have different characters. Sometimes spleen is shown, and sometimes not. So it's mimicking the natural one. And that's the story of the tissue-resident immunocytes. The next step is exosome specificity for target cells on in vivo studies were just done. Exosomes' role for CNS immune system interaction on sentinel lymph nodes for brain and spinal cord. In vivo, more unknowns now, and PET resolution allows. Thank you.
Thank you very much, Professor Lee, for your interesting talk about basic investigation you did. Thanks a lot.